Okay, thank you very much. I'm very happy to be here for the first time in Lodz, but not the first time in Poland. And what I then wanted to do today is to describe to you how we have attempted to sequence the genomes of our closest evolutionary relatives, Neanderthals and Denisovansons, and then spend most of the time on what we can learn from those genomes in terms of how they have contributed to the physiology of present-day people and how modern humans may differ from Neanderthals. But before we start, I just want to remind you, and please tell me if you hear me in the back. I don't know if the microphone works. It's okay. Let me just remind you before we start then that, as you are all aware, I think, modern humans appear in Africa and quite recently, less than 200,000–300,000 years ago, and start spreading out of Africa less than 100,000 years ago. But the interesting thing then is that modern humans were not alone at that time. There were other forms of humans around. Most famously then Neanderthals here in Europe and Western Eurasia, and other forms of now extinct humans in East Asia that we know less about. So Neanderthals to the left here then are these robust forms of humans. As you know, they appear maybe somewhere 400,000 years ago or so in the fossil record, and exist until 40,000 years ago, when they then disappear, generally in different areas in connection with that modern humans appear. And our lab has then been obsessed with Neanderthals, if you like, since 25 years or so. And you may ask, why should we be so interested in Neanderthals? And I think for me, there are at least two reasons for that. One is this fact that they are our closest evolutionary relatives. So if we want to define ourselves, all people on the planet today, from a biological or genetic perspective, it's really them we should compare ourselves to and say, how are we different from them? How are we similar to them? And another reason is that they were here quite recently, 40,000 years ago. So an interesting question is, what happened when Neanderthals met modern humans? They were here only, say, 1,400 generations ago or so. What happened? How are we related to them? But then, if you want to address this from a molecular perspective, you then need to retrieve the genomes, the DNA, from remains that are at least 40,000 years old. And there are lots of technical issues with that we have then worked on for almost 30 years in the lab. And those technical issues, as you can imagine, come from the fact that this old DNA you extract from bone is degraded to short little pieces of 30, 40, 50 nucleotides, relative to modern DNA that would be tens of thousands of nucleotides long. It's also very little of it there, often 100,000-fold, 1,000,000-fold less than in a modern tissue. And it's chemically modified, I should point out, that can induce errors in your sequences. And the vast majority of the DNA in an old bone doesn't come from, say, Neanderthal. It comes from bacteria and fungi that have lived in the bone over thousands of years. So one problem with this, in addition to these physical things, is that since there are so tiny amounts there, even small amounts of present-day human DNA that wouldn't at all show up when you study this modern DNA can overwhelm your experiments here when you study ancient DNA. But all these things you can overcome, all these technical issues, and then sequence hundreds of thousands and millions of short molecules, take these sort of modifications that exist there into account, and then map these fragments of DNA and see where they fit on the human genome to reconstruct, say, a Neanderthal genome. And today we then have 3 Neanderthal genomes of high quality that is of a quality similar to genome you would sequence from an individual today. And they come, one of them from Southern Europe here, from Croatia, 2 from Southern Siberia. And we will soon have more high-quality genomes. There are at least 4 other ones in the making in our lab. One of them actually comes from Poland, from Stonia Cave, that many of you may be familiar with, in southern Poland. So it will be the first high-quality Polish Neanderthal genome. So one thing that then became obvious when I started comparing these Neanderthal genomes to present-day people was that if you look in individuals today, and if we just take an example, chromosome 9 here, and each line would be one individual, and we mark in red segments of that chromosomes that are similar or almost identical to the Neanderthal genome. You will find that people, in this case people of European descent, carry segments here that are almost identical to the Neanderthal genome. So that then suggests that Neanderthal had mixed with ancestors of present-day people and contributed DNA to people who live today. And that then adds up, if you look in people in Europe or Asia, to about 1 to 2% of the DNA comes from Neanderthals. But what you will also see here is that often different people carry different segments of the Neanderthal genome. So you can also jump from person to person and see how much you can puzzle together of the Neanderthal genome today from people who live today. And that's indicated in blue down here. And that's at least 40, probably actually more 50, 60% of the Neanderthal genome that still walks around on 2 legs today, so to say. So if you like, Neanderthals are not totally extinct, they live on a little bit in people today. So one thing that also became clear then is that this contribution from Neanderthals you find in whatever populations you look at today outside sub-Saharan Africa. So in North Africa, Europe, Asia, Australia, no matter where you look outside Africa. So also in parts where Neanderthals have never existed. So the sort of idea about how that happens, that contribution, is that when then modern humans leave Africa, they probably meet Neanderthals early on, probably in the Middle East, and mixed with Neanderthals. And then when they then continue to spread over the rest of the world, they can sort of take with them this Neanderthal contribution also to regions where Neanderthals never existed. So it was another interesting finding that came out early on when we started to study these Neanderthal genomes, and that came from this fascinating site in southern Siberia, the Altai Mountains, in the Russian part of the Altai Mountains, in Denisova Cave, where the archaeologists have found this tiny little bone that they were very skilled, I think, to realize that it might come from a hominin. And it turns out to be the last digit of the pinky of a child, of a girl. And it was possible to determine a good quality genome from this, and we were then very surprised to find that the genome of this individual was not close to the Neanderthals and not close to present-day humans, but went far back to common ancestors shared maybe in 300,000–400,000 years back with Neanderthals. So it's sort of a sister group to Neanderthals, but a deep divergence here. So sort of realized you had found some new form of now extinct human that we needed to give a name to. And after much deliberations with Russian colleagues, sort of called them Denisovans, after Denisova Cave, this first site where they were found. Just like Neanderthals are called Neanderthals after the Neanderthal site in Germany. So you can then ask, have these Denisovans also contributed to present-day people? And indeed they have. You find no contribution in Western Eurasia, but wherever you look in mainland Asia, you find something like clearly less than 1%. maybe 0.2% or so of the genomes come from these Denisovans. Interestingly, in Oceania, so say Aboriginal Australians, Papua New Guinea, you have up to 5% contribution from Denisovans on top of this Neanderthal contribution then. So, and we can actually jump over this. So if we then summarize what we think we then know about the origin of Neanderthals and Denisovans and of modern humans from studying genomes, we then think that Neanderthals and Denisovans have some common ancestor in Africa, presumably. Probably this is what paleontologists might call the Homo heidelbergensis or so, that come out of Africa and then evolve in Western Eurasia to what we call Neanderthals, and in Eastern Eurasia to what we call Denisovans. The border between these groups we don't know, and it has shifted over time for sure, but we do know that at this site in the Altai Mountains, Denisova Cave, at sometimes there were Neanderthals there, at other times there were Denisovans. So sort of this is the eastern range of Neanderthals, and perhaps a western range of Denisovans, at least at some points in time. And then modern humans evolve in Africa, as you said, start spreading out of Africa and the Middle East seriously, say 50, 60,000 years ago. They mixed with Neanderthals early on, and it's now clear that they actually, as they spread, they mixed several times with Neanderthals. And in the East, then mixed with Denisovans. And it's also clear that they mixed with at least two different, rather distinct Denisovan populations here in the East. And modern humans then continued to spread out to areas where no hominins had been before. And Neanderthals and Denisovans disappear, but then live on in this contribution in present-day people. So one interesting thing that is happening these days is that we start to get very direct evidence of this mixture with Neanderthals. And the first case of that came from Romania, from this site, Oase Cave, where cavers then found back in 2010 or 11 a human mandible. It looks like this. It was carbon dated to approximately 40,000 years ago, and it looks like a modern human mandible, I'm told. So it's sort of one of the earliest modern humans in Europe that we have, that lived at a time when there might have been Neanderthals around. So we were very, very excited to look at the genome of this individual to say, have this individual already mixed with Neanderthals? So if we now look at this genome here, every chromosome is indicated, and in blue are regions very similar to the Neanderthal genome. You will see that not only have this individual Neanderthal ancestors, there are long, huge chunks of millions of base pairs, sometimes more than half of a chromosome arm is almost identical to the Neanderthal. So this, of course, indicates that this person had a Neanderthal ancestor quite recently in its family history. So you can then show that either 6, 5, or 4 generations back, and you're unsure of that because it's quite stochastic, of course, which part of chromosomes you inherit from ancestors back in your family tree. This individual had a Neanderthal ancestor. So we almost sort of caught someone in the process of mixing with Neanderthals. And recently then in Bulgaria, there is this site, Baciokiro, where one finds technology that's typical of the very early modern humans, sort of this initial Upper Paleolithic that come to Europe, and you find deep in the layers there remains of modern humans that are 45,000 years old. Higher up you find people that are 10,000 years later in the same cave. So in these deep layers there are 3 remains from which we have been able to retrieve DNA. And strikingly, all 3 of them have more Neanderthal DNA than people today, and have these sort of long segments on their chromosomes here. So you can show that all of them have Neanderthal ancestors a couple of generations back in their family tree. If we move to 10,000 years later in the same cave, they look pretty much like people today, about 2% Neanderthal DNA, and no sort of close family history there. So it's quite striking, I think, that in many of these earliest modern humans that one is able to study, you find that they have mixed with Neanderthals rather recently. They have Neanderthal ancestors quite close in time. So it suggests, I think, that the first modern humans that come mixed quite extensively with Neanderthals. And that maybe part of the story how Neanderthals disappear may not be that they were killed by modern humans or something like that, but maybe at least part of the explanation is that Neanderthals were simply assimilated into perhaps bigger modern human populations that come to Europe. Clearly, it can have been different scenarios in different regions, and it's a complicated thing, but part of it can be simply like that. If we're naive about it, we said there were 50 times more modern humans than Neanderthals, that would end up being 2% of our genomes today. Right? But it's not that simple, of course, but a sort of part of the story could be something like that. But what interests me particularly these days is then, what does this genetic contribution from Neanderthals have for consequences today for people living today? So genetic variants, something that changed in Neanderthals, were present in most or many Neanderthals, and then came over, say 50, 60,000 years ago to present day people. I just want to give you some examples of that to give you a flavor for what these contributions can entail. So the first example I want to illustrate has to do with progesterone, which you may know is a hormone that has many functions in the body, but one major one is that it's produced by the corpus luteum in the ovary after ovulation and prepares the endometrium in the uterus for a possible pregnancy. And what we're now interested in is the receptor for the progesterone hormone in the uterus and in other parts of the body. And this receptor has a variant, the gene has a variant that exists outside Africa. So whenever you see a genetic polymorphism that has this pattern, where it's frequent outside Africa and not present in Africa, or very, very rarely, it's almost always of Neanderthal origin, these variants. So this progesterone receptor was known to exist outside Africa and was observed by others to come from Neanderthals. And it's quite an important polymorphism since it's associated with preterm births, of having premature babies. And it had been speculated, for example, that it would have been a selective disadvantage for Neanderthals who were homozygous for this risk variant and for having premature babies. But something that is now becoming possible, that I think is fascinating, is that we begin to have so many ancient genomes from modern humans over time. So starting, say, 10, 15,000 years ago, going up to present day time. So we have thousands of genomes that many labs have sequenced. So you can then follow the frequency of such a variant over time and see how it has changed. And let's see if this little video then works. What we're doing here is then looking at the compilation of modern genomes starting 15,000 years ago. In red are the archaic genomes, gray are individuals who don't carry this variant, and in black the ones that carry the variant. In this time interval then, 15,000 to 14,500 years ago, and we now try to go forward in time and see, yes. And you will then see around 7,000 years ago how it almost explodes the frequency of this, which seems to make no sense. If this is associated with having premature babies that are at big risk of dying, of course, particularly in early societies, why would it increase in frequency over time like this? So that sort of stimulated us to go and look in present-day biobanks what this is associated with, this Neanderthal variant. And here we are then looking at the UK Biobank, where you have 450,000 individuals approximately with genetic information and medical information. And we are now looking at the modern version of this, so the non-Neanderthal allele here, and ask what it's associated with in present-day people. And you will then find that the modern non-Neanderthal variant is associated with bleeding early in pregnancy, with miscarriages, and it's negatively associated with numbers of full sisters, and actually also full brothers, but it's not sort of significant. So here, the modern variant is associated with early miscarriages. So the story seems to be that this Neanderthal variant, yes, it's true that it's associated with premature births, but it is also protective against miscarriages earlier in pregnancy and results in more live births. So we think that it is a trade-off. This Neanderthal variant protects pregnancies that would otherwise result in miscarriages. And the price you pay is that some of those babies are born early, but still, of course, have a chance to survive. And we begin to have an idea what the mechanism might be too, because if we look at people who carry this Neanderthal variant, they express more of the receptor. So this Neanderthal variant results in that you have more of the progesterone receptor. And you can, of course, imagine that having more of the receptor means that you have more progesterone effect, so to say, in your organs. And quite fulfillingly, in the last 3 years, there have been 4 studies where one has given progesterone to women that have experienced previous miscarriages. So say 1, 2, or 3 or more miscarriages. And that then reduces or increases the frequency of live births. So it sort of fits with the idea that if you give progesterone, so you have more progesterone effects, you sort of can save pregnancies that would otherwise result in miscarriages. And another way to do this might, of course, be to have more of the receptor. So that's an example where a Neanderthal variant is actually quite positive to carry today. And this is sort of typical of many actually variants, genetic variants of medical importance that are known since a long time, that they sort of one now finds they come from Neanderthals. So one involves the sort of metabolism of ibuprofen that many of us take when we have pains. And so if you take 400 milligrams of ibuprofen, the half-life in your blood of that is around 2 hours. But some people have a lot longer half-life, 9 hours or so with ibuprofen. And those are people who carry certain variants of a cytochrome in the liver that is very bad at degrading cytochrome, and is quite known in the medical community, this. And it turned out, when we looked at this year, that this variant is closely related to the Neanderthal genomes and clearly comes over from Neanderthals. So it sort of affects you if you take ibuprofen, for example, may do. What we are also interested in are things that we believe are Neanderthal specific. So things that changed here and exist in all Neanderthals, and as far as we know, not in present day people. And the example I want to bring is also done by Hugo Seeberg, who's a sort of driving person behind much of these things of Neanderthal admixture that I talk about. He is particularly interested in ion channels that sit in the cell membrane. And he noticed that all the Neanderthal genomes were sequenced, and one ion channel had 3 amino acid substitutions. And that's no other protein actually has 3 amino acid substitutions in the Neanderthals. And this ion channel is not just any ion channel, it's a sodium channel that sits in the peripheral nerve endings and initiates a sensation of pain. So we expressed this ion channel, the Neanderthal version and the modern human version, both in frog oocytes and in human cells. And for a certain sort of electrical stimulation, the red here, the Neanderthal version, seems to let through more current, to be more sensitive for a certain level of stimulation. And I'm not a neurobiologist, but one could sort of show that this is not because the channel opens faster, but it opens and the inactivation is less efficient. So it remains open for a longer time once it has opened. And you could then you could show that none of single amino acids have no effect, but 2 of these amino acids, intracellular amino acids, seem to have the full effect here. So we then looked in the UK Biobank again, and we thought that this was unique to Neanderthals and doesn't exist in present-day people. But now biobanks are beginning to get so big, so you find even very rare variants in present-day people. So among 360,000 individuals, there were actually 1,300 who carried this Neanderthal variant. So quite a low frequency, but anyway allowing you to study the effect of this in people that live today. So it has come over, but it's very rare. So the people in the biobank there have answered questionnaires about lots of things in their lives. So if we just pool all the questions that have to do with pain, no matter what pain it is, back pain, stomach ache, headaches, what have you, I think there was 16 questions there that had to do with pain. And if we then look at sort of what pain is associated with, this was the first time for me to be able to sort of play with so big population data. I wanted to know what is the biggest correlate with pain in your life. And as you get older, it's sad to say that it's age. The older you are, the more pain you report in your life. And that's trivial, of course, because you have more medical problems, you have more pain. But more relevant then, carriers of this Neanderthal variant here do report more pain in their lives than people who are not carriers. And you should relate that effect size to something. It is in this age correlation here as if you were 8 or 9 years older. So sort of 8 or 9 more years of pain in your life if you're a carrier of this. But it's rare, as I said, to carry this. So, of course, that doesn't mean that we can say that Neanderthals sensed more pain, because as you're very aware, the sort of sensation of pain is modulated in the spinal cord and very much in the brain. But it's still striking that people today who are heterozygous for this, have 1 copy of this, do report more pain in their life. Neanderthals were, of course, homozygous for this, had this on both their chromosomes. So it's sort of tempting to say that perhaps our view of Neanderthals as sort of insensitive brutes may be wrong. Maybe they were actually wimps and everything hurt when the modern humans arrived. So another area that sort of we have all been involved with in the last 2 years or so is then the SARS-CoV-2 and the corona pandemic. And as you know, as of last night, actually, then confirmed 6.5 million people dead in this, and that's probably a big underestimate, of course. And as you also know, one of the big questions with this virus is why some people who get infected have hardly any symptoms or very mild symptoms, and other people become very sick and even die from it. And you also know that there are known risk factors. Old age is a major one, male sex, diabetes, what have you. But all these risk factors aren't enough to explain why certain people become more sick than others. They explain a lot of it, but in the end doesn't explain the full story. So there is also a big interest in looking for genetic risk factors to become severely ill. And there's one big international consortium who look for host factors, genetic factors for severe disease. And we're sort of very peripherally involved in that a little bit. And their first results came out in the early summer of 2020. The first association study of risk of becoming severely ill. And it was very striking because there was one big signal on chromosome 3 where there was a locus then that increases your risk to become severely ill, require artificial sort of oxygenation or even die. And we were then shocked when we looked at that variant, those risk variants there, because they are very closely related to particularly one Neanderthal genome here and further to the others. Whereas all the protective variants, the other ones, are distant from that. So this risk variant had clearly come over from Neanderthals and exist in some people today. And it's quite a substantial risk. This is now data from the beginning of the pandemic when there were other variants around in Scandinavia. But so if you're not a carrier, were at that time not a carrier of this and ended up in hospital, you had a 7% risk of dying. If you were a carrier and were admitted to the hospital, you had a 13% risk of carrying, so almost a doubling of the risk. And if you look, for example, in people who are under 60 years of age and have no other risk factors, it's actually a 5-fold higher risk to die if you're a carrier of this. That's also trivial in a way. If you eliminate other risk factors, the genetic ones become more important. But to be a genetic risk factor, this is quite a big one. So it's of course very interesting to see this region where this sits then. What are the genes in there? Unfortunately, it's sort of not easy to understand. We and others are studying these genes in this region very intensely. If we understood why you have a bigger risk of dying if you carry this Neanderthal variant, we might also be able to treat the infection better or the consequences of the infection. But this is then the frequency of this variant. It's again outside Africa, as you see. You can then sort of estimate how many extra deaths you have had in the pandemic due to this Neanderthal contribution from the frequency here and the increased risk. And that's then at least 1, if not 2 million extra deaths that have occurred due to this admixture. I shouldn't joke about these things, but it is sort of like the late revenge of the Neanderthals. What is very striking, though, is the frequency outside Africa here that you'll see. You will see that this variant is absent in East Asia. It's absent in China and in Japan. It's very frequent in South Asia. As India and Pakistan, there are 50% of people are carriers. It's a sort of allele frequency 25%, but you have 2 copies, of course, so sort of carrier frequency 50%. In Europe, it's around 16% carriers. So something must have influenced this frequencies in the past. It's very unusual to see such a big frequency difference between East and South Asia. So you can speculate, of course, say maybe something, coronaviruses have been endemic in East Asia, so this has been selected against. It must have had some positive effect then in South Asia. So this is sort of a good example of that variants may have different, genetic variants interact with the environment and can have both positive and negative effects. And Hugo Seberg again have found sort of an example of that here. So if we look at this risk variant for COVID, located in chromosome 3 here, these are these genes one is particularly interested in now. If we now walk almost 1 million base pairs down the chromosome, there's another gene there that some of you may have heard about, CCR5. That is this gene that is a co-receptor for HIV. That helps the HIV virus to get into your cells if you're infected with HIV. So if you then look at carriers of this Neanderthal risk variant here, you will find that they express less of CCR5 on their cells. So that would then suggest that it might be harder for the HIV virus to enter your cells, because we know that people are homozygous deletions of this that don't have any CCR5. They are actually resistant to HIV infection. And indeed, if we then look in 3 biobanks, in the UK biobank, in the Finnish one, and the US one, at the risk of being HIV positive as a function of this Neanderthal variant here, is you have a reduced risk to be HIV positive. So this risk locus on chromosome 3, it does increase the risk for severe COVID if you're infected with SARS-CoV-2. But if you're exposed to HIV, it actually reduces your risk of becoming infected. So both positive and negative effects. So as so many things, it's sort of at least a double-edged sword. But as we've seen, there must have been other things in the past in East Asia and South Asia that influenced this. So as all of these things, it's probably actually a multi-edged sword. Before I leave the SARS-CoV-2, just to mention that there are also positive variants. One now finds other variants associated with severe COVID, one on chromosome 12 that comes from Neanderthals, and in that case it's protective. And very briefly, just point out that in that case one understands the mechanism very well. There are 3 genes in there that activate an RNase that degrades double-stranded RNA, so degrades intracellularly the viral genome. And this variant is also, this sort of genetic variant is also protective earlier on in the SARS-CoV-1 infections. So the influence from Neanderthals then on SARS-CoV-2 is sort of this doubling of the risk, at least, on chromosome 3. And a reduction of risk, but unfortunately a much smaller effect, like 1/4 of a less risk to become severely ill from this one. So finally then, what we are also then very interested in more and more are things that are unique to modern humans. So genetic changes that happen since we separated from Neanderthals and Denisovans and spread here to everybody today. And why are we so interested in that? I think that that's because we do think that modern humans are quite special in some way. That there might be, these variants may underlie some functions that are unique to modern humans. And we think they are special, of course, because with time, technology starts changing really rapidly with modern humans. We get art that are really depicting things. This is perhaps the most advanced, probably, Neanderthal art that we know about. With modern humans, art becomes much more frequent and really figurative. And modern humans, of course, spread over the whole world and over water where you don't see land on the other side and so on. So we can now, with the help of the Neanderthal genome, of course, list everything that changed here, that is not in Neanderthals, where Neanderthals look like the apes, and is present in everybody today, as far as we can say. So we can make such a catalog. And that list is not very long. It's around 30,000 changes in the genome. And if we just focus on one simple part of it, we sort of, to try to elucidate these functions, we've sort of been lazy and said, let's start with amino acid changes in protein. There are just around 100 of them. And you can list them here. And I wanted to, if I have 2-3 minutes more, 5 minutes more, just give you 2 examples of this. 1 or 3 genes here that stood out, also one of them has 3 amino acids, and one has 2, one has 1, that are all involved in cell division, in the spindle and kinetochore that pull the chromosomes apart at cell division. So it seemed totally surprising that something so fundamental as cell division would have changed recently in human history. So we speculated that this might have to do with, brain development. We have this bias to things that was special with modern humans. Is cognition in some way? That this might have to do with how neurons sort of evolve during development. And we then had thought back to a study where we had made brain organoids. So from stem cells, human stem cells or ape stem cells, you can make little balls in cell culture that mimic the early stages in brain development. And then look at the epithelium here, where cells divide down here, they migrate up here and form neurons. Comparing apes and humans, you find hardly any differences. But there was one difference we found, and that's Felipe Mora Bermudez in Wieland Huttner's group in Dresden that looked at this. And you can now see a little movie where you see the cell nuclei, the chromosomes here, the stained DNA, how they line up during cell division in a metaphase plate and are pulled apart. So if we follow this on the right, the chimpanzee, you see how it lines up in a plate here, and then the chromosomes are pulled apart there. And there in human, it happens there, and you saw that it happens slightly later. And one of the few differences he found between human and chimpanzee cell division is that this time when they line up here is longer in human. That made us then think about these changes again. So we engineered them into the mouse in these 3 genes, made a mouse and looked at cell division like this. And lo and behold, when we humanize the mice, their metaphase do get longer. And when we just put in 2 of these genes modified, they also get longer. When we put them in singly, we see no clear effect, and this gene then seems to be not involved, so we're sort of not so interested in that. So these 2 genes seem to have to do with this. So we then went to the human stem cells, engineered them back to the ancestral Neanderthal state, and we see this reduction then as we expect in metaphase length. So this metaphase is important for separating the chromosomes accurately. So if we then look at the numbers of lagging chromosomes that are missed when cells divide and start sort of not move to one or the other nucleus, that is increased in the ancestralized brain organoids here. These are very small numbers, it's not really significant, but it changes in the right direction. So it then seems that in these progenitor cells here, cell division is more accurate in modern humans than in Neanderthals. So we somehow seem to have more accurate cell division. The question is, what effects does that have? And that is sort of where we are. We're of course testing the mice now to see how smart they are and if they have some behavioral changes and so on. And I realized I don't have time, but the last example came out last week in the press. There is one metabolic enzyme here that also affects how brains how these stem cells here divide and results in more neurons higher up in the epithelium. But we will sort of skip that at the moment and just come to the end here, sort of saying that what we see in this list is then this gene here that increase the number of neurons, these 2 genes here that sort of make for more accurate cell division. So it's sort of suggestive that to some extent neurogenesis, some neurons are formed during development in modern humans differ in some quantitative and qualitative way to Neanderthals. And an interesting question for anthropologists, I think, is then, what does that mean? How is it compatible with what we see of behavior of Neanderthals and modern humans from the archaeological and the fossil record and so on? So that's sort of what we work on. There are a number of others of these genes who are also looking at the effects. But what I hope I have then done is to convince you about that having the genome of our closest evolutionary relative here is interesting, because we can figure out what changed on the human lineage, what changed on the Neanderthal lineage, what changed earlier back here, and study the effects of this contribution to present-day humans. And we do that then with the help of biobanks to a big extent, also by sort of editing these changes in the genome, in cells, and studying their effects, and sometimes then also humanizing mice and looking at the effects. And this work is done mostly in Leipzig, at the Max Planck Institute, but also particularly the mouse work in a small group we now have in Okinawa in Japan. And with that, I then thank you for your attention, and maybe it's time for one question or something.
Please join in. Thank you.